Hi, everybody. Thank you for coming to my talk. My name is Nathalie Alomar. I'm a graduate student with Martha Munoz over at Yale University, and today I'll be talking about trait diversity in Plethodon salamanders. Now, if you saw any of Martha's talk yesterday, this will be a little familiar, but I'm briefly going to go over these three key dimensions of diversity. We have species richness, ecological diversity, and phenotypic diversity. And these three dimensions can be increased or decreased across a lineage's history. For example, we can have a case where we have high species richness, high ecological diversity, and high phenotypic diversity, which is what we see in adaptive radiations. An example of adaptive radiations are these Caribbean anoles, where we really see them defined by their ecomorphs. So their morphology is tightly linked to their microhabitat specialization. So we can see kind of that crown giant up over there that has a morphology that allows it to occupy higher up in the canopy, compared to, say, that little twig anole that has a morphology that allows it to balance on those more narrow branches. Now, we have a counter case to adaptive radiations, right? And these are non-adaptive radiations, where we still have this high level of species richness, but this time pretty low ecological and phenotypic diversity. An example of this is the Batrachoceps salamanders in California, where we see this nice radiation, but this time pretty limited ecological and phenotypic diversity. And we can talk about how we can increase and decrease these axes in many different examples, but my work and my dissertation really kind of hones in on non-adaptive and adaptive radiations. And when we think about radiations, whether they be adaptive or non-adaptive, a lot of focus has really emphasized on the morphology of the organisms. So again, we can think of the really great morphology we see in the Caribbean anoles, or the lack thereof in the Batrachoceps salamanders. But still, a lot of the emphasis has been put on the morphology. And this emphasis on one phenotypic trait can sometimes neglect the fact that species are radiating along multiple phenotypic axes. And the patterns and rates of evolution might differ depending on which trait that you're looking at. So essentially, we can have divergence in one phenotypic trait be really fast and more labile compared to another phenotypic trait where it might be inert. And we have seen this happen before, even in our classic case of adaptive radiations, the anole lizards. So here, if you don't know about lizards, they like to thermoregulate, they like to maintain their preferred temperature, and at low elevations, they do this by basking on tree. They go into the tree, they get warm, and then they're good. However, at higher elevations, basking on trees, it's not sufficient enough for them to maintain that preferred temperature. So instead, they go and bask on the boulders are hotter, and they're able to maintain that preferred temperature. And this shift between basking on trees to basking on boulders has two different consequences on the phenotype depending on where you're looking. When you look at the physiology, we see a slower rate of physiological evolution, right? So that light curve in the bottom hand corner is going to be the high elevation species. The darker one, if you can see it, is low elevation. And this is a thermal performance curve with temperature on the x-axis, performance on the y. And what we see is that the CT max, or the heat tolerance, and the thermal preference are really similar between low and high elevation species. The CT min is different because these are higher elevations. It's more homogeneous at night. But overall, we see the slower rate of physiological evolution. But this is not the case across the whole phenotype. When we look at their morphology, we see this faster rate of morphological evolution. They have these flatter skulls, shorter hind limbs that allow them to climb up into these rocks. So essentially, we see that there can be both stasis and divergence in different trait dimensions. And this project really asks these bigger questions. Are there any quote-unquote hidden dimensions of phenotypic diversity? And to look at this, we're looking at Plethodon salamanders, who I think are one of the most adorable animals that we have. You can find them in the Appalachian Mountains, where it's like the hotspot of salamander biodiversity. They're terrestrial amphibians. I have loved working them in the last couple of years. They can be broken into three main sublineages. So the red node is going to be the Plethodon cinereus complex, also known as the redback salamanders, really common along the eastern coast of North America. We also have Plethodon welleri and Plethodon glutinosus in the green node, commonly known as some of the slimy salamanders. And one thing to note about these salamanders is that they are highly cryptic species. So I have 6 photos of salamanders on the board, and if we were to assume which ones are their species, we might think that the first row is species 1, the second row is species 2. But what we actually know is each of these photos show 6 distinct different species, just with really minimal morphological variation. And this kind of plays into this bigger role of Plethodon salamanders. They're considered this classic case of non-adaptive radiation. They have very minimal morphological variation. And this is something that this project really wants to dive deeper into, right? So we know that they have this nice radiation, but there's a lot of things about these salamanders that are really unique, right? They are terrestrial, they're direct developers, they're woodland, and like most members of their family, they are completely lungless. So what we really wanted to do is dive deeper into their physiology, especially because most of the physiology that has been done in this clade has been within just a few individuals, but never across the whole clade or between and within sublineages. And that's really what we wanted to tackle with this project. So we're asking the question, is ecophysiology a hidden dimension of phenotypic diversity? And is plethodon physiology more diverse compared to their morphology? So to do this, over the last couple years, I've had a really fun time going across the eastern United States collecting salamanders with a bunch of help from a larger team and also a lot of undergrads. When we go out to collect them, we're getting environmental data, including relative humidity, dew point, wind speed, the elevation, air and soil temperature, and also the substrate that the salamander is found in. After that, we take them back to Connecticut with, again, a bunch of help from undergrads. We're looking at skin resistance to water loss rates, how good they are at retaining their water, their metabolic rates, critical thermal minimum and critical thermal maximum, which again is just going to be the heat and cold tolerance when we're looking at the thermal performance curve, and this is related to the locomotion, so when they're not able to right themselves. And then we're also looking at thermal preference, which is the temperature the animal prefers in the absence of any ecological opportunity. We also have morphological traits that we have from a database that we're looking at, and of course we're collecting mass that we can use to correct for some physiological traits. So let's get into some of the results that we have so far. And I'm going to be showing a number of cont maps here, and the higher the value is going to be a redder or warmer color, and the lower value is going to be a bluer or cooler color. And this is going to be CT max, which is at heat tolerance. And what we can see here is pretty minimal variation between the different sublineages, but some slight variation just across the whole clade, right? So they do tend to be around 32.5 degrees Celsius for their CT max, and we have some variation just across the whole clade. And we see a similar pattern when we look at cold tolerance over here. Again, some good variation across the whole clade, so they have a more diverse physiology in their cold tolerance, but between sublineages, not so much. Now this is the thermal preference, and here we start to see a little bit more of a pattern where the glutinosis group is starting to prefer these warmer temperatures compared to the Wellerii and the cinereus group, which are the blue and the red nodes. When we look at the metabolism, and this is the residuals of mass and metabolism, taking account to the size of the organism, we see that the glutinosis group, again, is generally having a lot of variation, but have these higher metabolism compared to the cinereus and Wellerii group. And then when we look at the skin resistance, we're seeing that they're having lower skin resistance for the cinereus and Wellerii group and higher skin resistance in the glutinosis group. So this is really cool for us for a number of reasons. We're starting to see some differences between and within sublineages, and we really wanted to see if this is correlated with any of the microclimatic temperatures that we got when we were in the field. So here I have another graph, and I really want you to focus in on that that black box right there. In this graph, temperature is going to be on the y-axis. The x-axis is vapor pressure deficit, which is essentially how dry the air is. So a higher value is more dry. And basically, in that black box, those little squares are all members from the plethodon glutinosis group. So we really find them at these higher temperatures and at these most dry conditions compared to the other sublineages, and that could be why we're seeing these higher thermal preference, higher metabolism, higher skin resistance to water loss rate. But if you remember the beginning of my talk, I was really interested in how these traits are evolving through time. So we've also looked at univariate analysis of these different traits. So this is the hydric rates of evolution, those skin resistance to water loss rates. And we see that the glutinosis group right there, the green node, and the Wellerii group are having these relatively higher rates of evolution. And within the glutinosis group, we have another clade that's really driving these higher rates of hydric evolution. When we look at their metabolism, we see something a little bit similar, but this time we see that it's primarily being driven by the glutinosis group, having these higher rates of metabolic evolution. Now, this is really fun to see how these patterns go when you're looking at the rates of evolution between sublineages, but now we want to compare it across all of our traits, right? We have hydric, we have thermal, we have metabolic, and also morphology. So then we've done a multivariate rate analysis of seeing how all of these traits are evolving, and when we look at that, we see that those first three kind of groupings are all going to be physiological traits. So blue is hydric, green is metabolic, and red is thermal. And the gray one is going to be our morphological traits. So when we look at them all together, we see that the physiological traits are evolving faster than the morphological traits, which is essentially the opposite of what we saw in the Caribbean anoles. So what is this project telling us, and what are kind of the key takeaways? The first one is that the plethodon physiology is fairly diverse, right? They kind of have this bad rap at being these non-adaptive radiation, pretty lackluster species. And they are when you look at their morphology for the most part. But when you consider their physiology, you're seeing a different part of the story, and that they actually might be more diverse. And we're really excited to kind of dive deeper into that and see other ways that these patterns might be showing up. And we're seeing that their physiological traits are diversifying faster than their morphological traits, having this faster rate of evolution. And overall, this project has been really fun to kind of think of the ways that the potential biases that we have when we're thinking about a system might change the story that we're telling, right? So with this project, it really has forced us to take a step back in the way we think about non-adaptive and adaptive radiations, and especially what do we mean when we say phenotype? Is it just morphology, or is it true the full phenotypic axis of an organism? And then, of course, I want to thank all of my funding sources, the lab, my collaborators, and really, there's not enough room for all the undergrads that have helped with all of this work, but a really big emphasis on my undergrads that have helped me. Thank you.
I probably will have an opportunity to ask you questions later, but just for now, I'm just curious about the communities of these salamanders, and do they tend to be made up of like the different clades in one region, or does each clade sort of have a different region geographically?
Not necessarily. So a lot of them overlap a lot in their regions. So like I said, the Appalachian, especially that North Carolina area, you can see a lot of overlap in like the glutinosis, the cinereus group. So they have a lot of overlap in the area, but another previous study has shown that even within this overlap, you're seeing these microhabitat preferences that these organisms are showing even in some overlap in that area as well too. Yeah, thank you. Okay, thank you.
My name is Mayara Neves. I'm a postdoc at Corey Evans' lab at Rice University in Houston. And in our lab, we are really interested to understand how traits evolve. And in the specific project, I will try to understand deeply how living in extreme environment affect the school evolution of the ice fishes. So in this project, I already would like to say thank you for my collaborators, in you all here to listen to my talk. In a broad perspective, across the vertebrate, there are a lot of examples of adaptations for living in different environments, in different kind of habitats, like deserts, caves, deep sea, and all of these kind of habitats require adaptations for different levels, like biomechanical, physiology, morphological, and other ecological aspects. So therefore, this kind of adaptations has huge impact on phenotypic evolution. Considering that, one of the most interesting questions it is if living in extreme environment constrain or accelerate phenotypic evolution. And there are a lot of examples, empirical results, tends to indicate that the adaptations for extreme environments can constrain and also accelerate the rate. So there is not a pattern exactly for vertebrates. So considering that, not just the tempo of evolution, but also the mode, it's another important raising question. It is about how these adaptations evolves, if it is via modularity or integration. Briefly, morphological structures can be considered as modules, like here, dentary and articular angular. If you are not a fish biologist, you probably have difficulty to understand what is these bones, but basically this lower jaw in vertebrates. And thank you for Howard, because he produced this wonderful gift to help you to understand what is this kind of systems. So in integrated systems, both modules or bones can change their shape together across evolutionary time. In modular system, each module or each bone evolve independently. So considering these aspects are really relevant to consider when we want to understand the adaptation of constraint in evolution. And to test this kind of predictions and try to understand better these patterns, we investigate how living in extreme environment affect the morphological diversification. So for that, we focus on notophineoids, a superorder of perciformes with around 155 species distributed in 8 families, and the most specious family are considered cold fishes and barbel plunder fishes. As you can see, the notophineoids with 56 fishes, and second, Arcted draconidae with around 35 species. As you can see in this image, some of these families have a geographical distribution restricted to Antarctica, associated with the Antarctica polar front, while others families, they reach out the south of the South America, so they have a broader distribution compared to other families. Considering that, the adaptive radiation of notophineoids relates to Antarctic circumpolar current formation. Well-established studies have been conducted on their morphology adaptations, such as antifreeze glycoproteins, lack of functional erythrocytes, and reduced bone densities. However, considering the fundamental aspects of the skull related to food intake, breathing, and the number of bones, we look deep into the evolution of their skull shape. So that we have 3 main goals. First, then, to investigate the skull shape diversification, compare both notophineoids and other perciforms. Second, to estimate the rates of evolution in this group. And third, to analyze the mechanism of morphological adaptation in ice fish. So to answer these questions, we use a morphological data based on micro-CT scan, a huge data set with 174 species, and specifically notophineoid comprise 82 species, and other perciforms with 92 species, and we use the Harabowski phylogeny to test these evolutionary aspects. Regarding the methods, we, after scan all of these species, we segment the species in a mirror and digitize the landmarks in checkpoint using a scheme with 145 landmarks scheme, and analyze the data in R. So as you can see here, our first result, we can observe the difference between these 2 main groups, other perciforms who are not restricted to Antarctica, and notophineoids, the species who are geographical distribution restricted to Antarctica. And if you are not familiar with osteology of fishes, you can see a little bit these bones apart to understand how the shape of these bones are a little bit different between these 2 groups. So it was the first visualization about how these 2 groups are different in terms of shape, not just the entire skull, but also each bone independently. So with that, for the first goal, we investigate the morphological disparity using phylomorph space in PGLS. We estimated the rates of skull shape evolution on base rate, considering the Brownian model. And for modularity integration, we use 2 methods. First, covariation rates and graphical modeling, considering 7 hypotheses. First hypothesis, all bones separately, and 4 functional hypotheses and 2 developmental hypotheses. After this, our first result relates with morphological diversification. We can see this huge pattern, as you can see in other groups, when you have the most elongated skull versus shortened skulls. Here, in terms of notophineoids, you can see a high disparity in this group, especially in this X1. So we have a notophineoids with skull similar with other perciforms. So we can see a lot of blue dots mixed in these other gray dots together with other perciforms. However, we have another opposite side with elongated skulls, which makes really interesting because we also have some other perciforms with elongated skulls. So looking deep into the feminine aspects, who are these guys with elongated skulls? The species with elongated skulls belong to mainly to 2 families, Bathydraconide and Canictide. The families who has more histological, morphohistological adaptations, morphophysiological adaptations, like antifreeze glycoproteins. Meanwhile, the other families included the most diverse family. We have specifically this skull, short skull, together with other perciforms. Seems we have a huge disparity in terms of skull. So why we can see more about them families? I analyzed at least two main drivers so far. First, region, because in this plot, if you see the phylomorph space, we can see some main drives that can explain the morphological disparity in notophinioid. Notophinioids who are restricted to Antarctica, they can have both short and elongated skulls. However, the species that are not restricted to Antarctica, in the left side of the graph, they have just short skulls, while other perciforms, they have some case of convergence with elongated skulls, but without geographical restrictions to Antarctica, like notophinioids. So, if a region is not the main drive that can explain this disparity, I look deeply in habitat. Given the fact the region is not the only main factor, we can see generally deep demersal and shallow pelagic species have elongated skulls, while species that live in complex habitats like freshwater or live in coastal regions, they have shorter skulls. Also, it is interesting how some freshwater species have some case of convergence as well. In this sense, we can predict that if you are a fish and you live restricted to Antarctica and in a deep demersal habitat, probably you have a elongated skull, right? So it is a combination between region factor and also habitat. And if you look at the results about morphological disparity, here, independently of the family or the habitat, it seems the region it is the main factor to play a relevant role in morphological disparity. Considering the rates of morphological diversification, we can observe some fast rates of skull shape evolution in both notophinioids and other perciforms. So above of the three, we have families belong to notophinioids, and this another second huge yellow-red group are another families of perciforms. And what we can see exactly when you look to notophinioids. When you give this zone in notophinioids, the fast rates are observed in the three main families with elongated skulls, and that ones who live in deep habitats, like Canictide, which has antifroze proteins again, just remember. These fast rates are associated with family diversification and seems to slow down after that. This is interesting because match with the complete formation of Antarctica circumpolar current. Other notophinioids have similar skulls as other perciforms, and they don't have a considerate fast rates of shape evolution. So they are look at other perciforms in terms of not just in shape of the skull, but also in the tempo of evolution. And probably you are curious, why other perciforms has rate of evolution? Who are these guys in this part of the tree? Well, I call them perciform strange things room because they are really, really strangers in their, not just in terms of a skull, but the entire body. They have really weird morphologies and also occupy different kinds of habitats. So the difference here, it is because they have a continuous and fast rate across the time. So in opposite to notophinioids, they have a long evolutionary history. So this is a combination of tempo and modification across the time. really profound when you compare to the notophenoids, which has really recently adaptative radiation. Regarding modularity integration, our modular test indicates that the skull shape evolves via integration, so not that the bones doesn't change independently, but change together. With functional 1 and 3 are the best models. And specifically, if you see the rates of evolution in terms of all bones, we can see neurocranial and maxilla with the highest rates of change when you analyze this kind of rates of evolution for each bone separately. And my main message for you today, after this huge effort, it is, first, there was an accelerated pace of morphological evolution within the 3 icefish families, and this matched with the adaptation time. And increased integration could be potentially enhance evolutionary capabilities, and integration can facilitate their morphological evolution and subsequent adaptative radiation in Antarctica. And all of this information is really important because, as you know, we are during the time of climate change. What will happen with these species who evolve and adapt really fast during a short time in Miocene? So it's a way to think more about what will happen with these species. With that, I'd like to say thank you again for my supervisor, Carl Evans, Haon Chen, our grad student, and also all the museums who send the species to scan across this time, and for the funding, and you all for the attention. Thank you very much. Yes?
Really interesting talk. Do you know if there's any plasticity within that species? So like a shift from a complex environment to a more shallow environment, will that induce a shift in...
Okay, you mean this, if some species can change the environment?
Yeah, does that lead to a shift in facial shape?
I don't know exactly if there is, but since I know they are more restricted in terms of habitat. So because for demersal fishes, they also have another morphophysiological adaptations. For example, their bone density are really low densities. So basically, they have buoyance variations, but they don't change a lot. But some species has possibility to change in water column, but yeah, basically they elongate. I guess they are more restricted in terms of habitat as well. Thank you. That's it.
Hello everyone, my name is Rohit Kapila. I'm a postdoc at Florida International University working with Dr. Jenna Pierce. We work on these tiny worms called C. elegans. So I warmly welcome you all to my talk today. I'll be talking about mutational makeover and how SNPs and structural variation dynamics change over across 3 different strains of C. elegans. To give you a little background about the model system, C. elegans is an androdioecious species where there are very few males and a lot of hermaphrodites. The preferred mode of reproduction in them is self-fertilization, and the number of hermaphrodites, the number of males at times in the laboratory strains can be as low as 1 in 1,000 individuals. So one can question what is the role of the males in these populations. Well, studies have found out that the frequency of male increases in response to any kind of chronic stress. For example, in this paper in 2022, they showed that when there was chronic irradiation, the male frequency increases in the population, which suggests a possible role of males in promoting population's resilience. I try to answer the same question, but from the other end, asking, does more males makes you more resilient to some kinds of stress? To answer that, I work with 3 different strains of C. elegans. One is N2, one is CB4856, and the other one is called AB1. N2 has the least number of males in the lab, 0.1 to 0.2%. CB4856 usually have 1% of male in the population, and AB1 has somewhere from 5% to 20% males in the population. What I did was I took these strains, mutagenized them for consecutive 5 generations using 2 different mutagens. One is EMS, that is known to cause point mutations. The other one is formaldehyde, that is known to cause complex lesions in the genome. And I, after that, allowed them to recover from these mutations for 3 consecutive generations. And then I measured their relative fitness before they were mutagenized, after they were mutagenized, and after they recovered from this mutagenic abuse. With an expectation that the strains with the maximum frequency of male would be best at recovering from the mutagenic abuse, which is AB1 in this case, which has the maximum frequency of males. CB1 would be better than N2s, which have the least males, and worse probably than AB1, which has the most number of males. This was the expectations. But to my surprise, that was not the case. What you see on screen right now is the relative fitness on y-axis in 2 graphs. One is for formaldehyde and one is for EMS. If you look at the x-axis and compare ancestors versus recovered, you would realize within 3 generations, almost all, in all the cases, the relative fitness came back to the same level. So irrespective of the male frequencies, inherent male frequencies that these strains had, they came back to more or less the similar level, which made me think probably more males might not mean more resilience in this case, but then I realized that there is more to resilience than just fitness resilience. There is genomic resilience as well, because every genomic change might not lead to a fitness-associated change. So I decided to look at genomic resilience in them. To put more context to this question, with change in male frequencies, the outcrossing rates also change. And we know that with change in outcrossing rate, almost every attribute of genome changes. There is an increase in genetic diversity, there is reduction in mutation load, there is increase in genomic stability, and the list goes on. And since I was working with mutations, I decided to look at the mutation load after they recovered from the mutagenic abuse, how much of the mutations retained in their genome. For that, I looked at both single nucleotide polymorphisms and structural variants in these populations. A lot of mutational studies have been built around, mutational theories have been built around single nucleotide polymorphisms because it's easier to get short read sequencing, which is cheap and very accurate, whereas studying structural variations would require you to have long read sequencing, which is relatively more costly and it's more error prone. To get the best of the both worlds, we got both short read and long read sequencing done on these worms. And we asked the question if mutagen purging ability is reflected in the male frequencies in these populations, and if it differs between SNPs and structural variants. And this was the bioinformatic pipeline that I used. We used long read sequences and short read sequences. We used their respective reference genome to align the sequences, and then using respective variant caller for SNPs and structural variants. We called SNPs and structural variants, and then we combined everything in Survivor to get best of the both worlds from short read and the long read data. And we were happy to see that we were able to capture very long structural variants as well for both EMS and formaldehyde across all the 3 strains. As big as 10 to the power 7 was the size of the structural variants that we were able to capture, starting from the small ones. So what's happening at the mutation purging ability level? So a lot is happening in this graph right now. I'll walk you through it. So these are the de novo SNPs and structural variants post-recovery from mutation for all the 3 strains. Every circle is for SNP, and every diamond-shaped figure is for the structural variant in the genome. The green ones are the N2s, which had the least male frequencies. Their efficiency to get rid of SNPs and structural variant is similar because, as you can see, for both EMS and formaldehyde, they are next to each other. But when we went to CB4856, which had 1% male, they were able to, their efficiency to remove structural variants was better as compared to SNPs, and it was opposite for AB1, the strains that had maximum number of males. They were successfully able to get rid of most of the SNPs, but structural variants was difficult for them to get rid of their genome after recovery for 3 generations. So the takeaway from this graph is that strain with most number of males retain most number of SVs, and there are strain-specific differences that we observed in getting rid of SNPs and structural variants. All the 3 strains dealt differently with structural variants and SNPs. To get rid of the strain-specific variances, what we are doing right now is we are evolving each of this strain into, experimentally evolving them into obligate outcrossing and obligate self-fertilizing using FOG and ZOL mutation. What FOG does is it kills the sperms in the hermaphrodites, making them essentially a female. So they have to mate with a male to reproduce, and Zol mutation makes them obligatory self-fertilizing by killing the males from the population. So we are evolving them in these two regimes, and at every generation we are providing them with the same mutagenic abuse using EMS and formaldehyde. And after certain amount of generations, we have decided 20, we are going to do again a fitness assay and a genomic analysis of similar kind on these worms to see if outcrossing in itself can make a difference in the mutation purging ability of SNPs versus structural variants, because what we have found so far, the strains are dealing differently with both of these kind of mutations. So conclusions so far from this ongoing study is that 3 generations of recovery from mutation was good enough for all the 3 strains to get back their fitness level to what the ancestors had, irrespective of the inherent male frequencies that they have. Strains with most number of males found it difficult to get rid of structural variants, and there are huge strain-specific differences in how these strains are dealing with SNPs versus structural variants, and probably having a mutagenic theory about increase in outcrossing reduces the mutagenic load might not be true if we consider structural variants as well as in addition to SNPs. I would like to say thanks to some of my lab members, Gabe, Saran, and Rahul, for conducting the fitness assays, my PI, Dr. Jenna Fierce, for teaching me the bioinformatic analysis that I did, Tori and Danny for helping me wherever I needed their help, and all the funding agencies. I'd be happy to take your questions and suggestions, more importantly. Yeah.
Changes in the male frequencies after mutagenesis, and were there any correlations between SNPs and structural variants and the male production frequency?
So actually this is the assays that we are conducting right now. We were so far working under an assumption that the male frequencies will go up after mutagenic abuse, but we never looked at the male frequencies. One of the hypotheses that we have is that probably everybody comes down to 50%. Whenever there is mutagenic abuse, everybody comes to 50%, and it's more easy for N2 as compared to AB to reach to 50%, and that's why there is not a lot of difference in the relative fitness at that level. But that's one of the working hypotheses right now, and we are conducting that assay right now. Also, if you work on worm evolution, we are planning to have a dinner tonight with every person from Worm Evolution. I think Jordan has sent out an email. We will be meeting outside the convention center, so you all are welcome. Yeah.
All right, so everyone, thank you for coming here. So my name is Cheng-Ruei Lee, and I'm from Taiwan. So I think all of you have been hearing a lot about the local adaptation and biodiversity in wild plants and animals, right? So today I hope to convince you that even for crops, they do have local adaptation and they do have variation, and these are very important for their spread, actually. All right. So we're humans and we eat things, right? But does it ever occur to you that what we eat, where did it come from, and how they originated? So archaeologists, they actually have some answers. So they call this process the food globalization, and based on archaeological records, so they have shown that things were first domesticated in different regions of the world, and then after that they started to spread, and they go this direction and that direction. Now the question is why did they go that direction or this direction? And is there a specific direction? And if there is a specific direction, why is that? Right. So in order to solve that, we actually work on a crop that we call mung bean. And mung bean, well, as I have in the first photo, they're the little green bean kind of thing. So they're like the Asian counterpart of common bean, actually. Right. So we knew that mung bean was first domesticated in India. So that came from archaeology. So what we did is that we actually worked with the three largest crop stock centers of the world, and we got more than 1,000 sessions. So what we did is that we genotype them, and then we run some and mixture and PCA stuff. So you can see that in the world, there are actually 4 major populations, right? So we got a Central Asian population, one from South Asia, one from Southeast Asia, and one from across the whole northern parts of Asia, right? Okay, so the first thing we look at is whether different stock centers have different proportions of these collections. So as you can see here, the first center, the Vavilov Institute in Russia, I mean, this collaboration was before the war, actually. So the Vavilov Institute is actually enriched for the more northern genotype, right? So they have the blue and the orange part. And then for the South Asian-based World Vegetable Center, they are more enriched in the Indian type. And also for Australiac Center, they're enriched for the Southeast Asian type. So this is saying that despite that, people think like maybe 50 years ago, there's a worldwide trend of seed stock exchange. But despite that, every stock center still have their own unique collections. So it's important to use all of them. Right, so the first question is, after they were being domesticated, how did they spread to the world, right? So we could imagine, well, maybe humans just bring them all across Asia, right? So to solve that question, the easiest thing is to just look at the phylogenetic tree. And from this tree, just look at the color. So you can see that from those black lines, they're the wild progenitor. So from those, we got the yellow South Asian type, and part of that becomes the light blue Southeast Asian type, and part of that becomes the darker blue East Asia, and then part of that becomes the Central Asia type, right? So this is the order of population divergence. And then the order is actually also very similar if you look at the population level tree, right? So this is a population level tree based on tree mix. So all of these results are consistently point to this very unique or strange direction of spread. So they do have a very strange counterclockwise kind of spread. And despite that, Central Asia is geographically closest to South Asia. Mangbin did not first disperse from South Asia to Central Asia, right? So we want to understand why. But before that, we actually did a lot of population genetics analysis to show that this trend is actually correct. And also this trend is also supported from linguistics, actually. Also, let me teach you a bit of Chinese. So in Chinese, there's a character that we call this hu, right? Hu. And then whenever you see this character, that means something originated from Central Asia and spread to East Asia. Because for the ancient Chinese people, when they live in East Asia, they call the people from Central Asia the Hu people, right? So which crops start with Hu? So you can see basically a lot of them. So we got pepper, cucumber, sesame, carrot, and everything. So we knew that they were being domesticated either in this Middle East or Europe. But despite that, what's the Chinese name of mung bean? We simply call this green bean. All right, so it is not hu at all, it is just green bean. All right. Now, so the linguistic evidence actually supports our idea that mung bean did not spread to East Asia through Central Asia. And then also notice these three crops, we know that they were being domesticated in India, and their name suggests that after they were domesticated, they can spread to Central Asia and then go to East Asia, but not mung bean, right? Why is that? Now, of course, the easiest idea is that there's a geographic barrier, right? So between Central Asia and South Asia, we know there is a Hindu Kush mountain, so maybe that's restricting the natural spread of those plants. Now, but remember, we are actually working on crops. So the difference between crops, the cultivar, and the wild genotype is that you can see that on the upper right-hand side, when a crop, a cultivar, when its pod mature, the pod will not open. So this ensures yield, and that's one of the most important domestication traits in legumes. And that also means that they cannot disperse on their own. So their dispersal totally depends on human. So the correct question to ask here is whether Hindu Kush, is it really a barrier for human seed exchange? So that instead of biology, we start to look for evidence from more history, actually. So we knew that long time ago, we knew the Aryan story, right? So they actually entered or invade India from Central Asia. So they cut through the Hindu Kush. So people go through Hindu Kush, and Alexander the Great himself also cut through Hindu Kush. And then there was a branch in the Silk Road that also cut through the Hindu Kush, and also these crops, they originated in India. They were first spread to Central Asia before they go to East, right? So Hindu Kush is obviously not a barrier, right? So since it is possible for ancient humans to actually spread the seeds all around, now the question is, why can they not establish in Central Asia, assuming that people have been trying to bring them to Central Asia? So one obvious reason is environment. So we started to do the ecology kind of thing, the environmental niche modeling, and you can see that, well, of course, like every group has their own most suitable region, but if you look at the region overlap, you can see actually there is a lot of overlap in niche between South Asia and Southeast Asia, but not between South and Central Asia. So it is easier to show in number. So we use the Schoeners-D. So 0 means no overlap and 1 means complete overlap. So imagine you were a Hmong and you start in South Asia. So it is easier for you to establish to go to East, right? And then it is very difficult for you to go to North, to Central Asia. Why is that? Because it is too dry. So we show that the precipitation is actually one of the most important factors. Right, so we have population genetics, we have niche difference now, but we need some proof, right? So our hypothesis is that maybe environment is so different, so Indian mung bean cannot be grown in Central Asia directly. So if that is the case, why do we still see Central Asian mung bean there today? Then that is suggesting that maybe in the history, like human may have changed their phenotype, so that's allowing them to be grown in Central Asia. Well, in other words, local adaptation. So we started to ask our collaborators for data. So since these are like crops, right? So agriculture people have a lot of field data. So the first thing you see here is their flowering time, and then we put them in. We got data from 3 different gardens, and then the 3 different color bars are 3 different populations. They came from South Asia, Southeast Asia, and Central Asia. So the first thing is flowering time. You can see that no matter where we grown them, the Central Asian population, they always flower the fastest. Right, so this is flowering time. Now, in terms of yield and in terms of the seed size, is the South Asian type have the largest seeds, which makes sense because they live in like a very wet area. And in terms of the yield component of pod number per plant, is the Indian type that has the most pod number per plant. And in terms of plant size, actually, it's actually a Central Asian population that's smallest. Right, so what does this all mean? So if we combine this is saying that the Central Asian genotypes, they're smallest, and they flower the fastest. But there's a trade-off. Once they flower, they cannot produce a lot of pods, or they cannot produce very large seeds. So this is a typical, like the fast cycling and lower yield kind of plant phenotype, right? And in plant ecology, this is a typical trait of drought escape. So because in Central Asia, the growing season is too dry, so they need to grow very fast, flower very fast. But the trade-off is that the yield is low, right? So so far so good, but somebody once argued that, by that somebody I mean reviewer. So somebody once argued that. So maybe the trade difference that we see today, maybe it was just the result of the past 20 years of breeding, right? So does this difference really exist like 1,000 years ago? So we do not have time machine, but luckily, no, we still don't have the time machine. But luckily, somebody actually did this observation for us. So what you see here, it was an ancient Chinese emperor, and then you could see the heart on the map, right? So that's where he lived. So he lived in northern China. So what did he say? So there's a record, and the record, let me translate for you. It says that emperor cares about agriculture. So first, he heard rice from Vietnam is drought tolerant, so he sent somebody to get the seeds into China. But then what's important are the sentence in red. That is basically saying that for him, for the guy that used to the mung bean in the northern China, he heard that mung bean in the south, especially India, they produce a lot of seeds, and the seeds are large. Right, so this is a 1,000-year record, and the phenotypic difference actually fit what we observe today. Right. Okay. So in addition to the farm phenotype, we actually also started to do some trait differentiation in terms of their root development under the normal or drought condition, and for the sake of time, like these are just the measurement of the shoot dry weight and root dry weight of these different populations, but these do not really show why this orange Central Asian population is different. What's really important is this trait. So this is the root shoot ratio, and then you can see that when these plants were grown under drought, on the right. Under drought, they have enriched root shoot ratio, so they grow a lot of roots, trying to absorb water, and they reduce the above ground part. Right. Now, despite that, you can see that the orange population, they almost always still have the higher root shoot ratio than the two other populations. Right. So, well, that's all of my talk now. So I believe that I have convinced you that there is local adaptation in crops, right? So they're crops, but they don't always look the same. So you may be familiar with this, the scientific method, and I'm a big fan of this, right? So first, we did some observation. So we have some population genetics. We see like what are the groups, where are they, how did they spread, when did they spread? And then our question is why did they have that specific spread pattern? Now, well, hypothesis, the first is that Hindu Kush is a geographic barrier, which was rejected by thousands of years of human history. And then maybe there is environmental adaptation. So we tried to do the test with all these stuff, and conclusion is, yes, despite we might think the crops are all the same, actually there are local adaptation. And these local adaptations are actually shaping their phenotype and are restricting how they spread. And actually, eventually, they may affect how different people in different regions of the world, how they actually eat their food, right? So with that, thank you very much.
So I was wondering actually about when you cross the populations, like Pakistan and Taiwan, do you see like outbreeding depression or heterosis? Any evidence of that kind of differentiation?
Good point. So they're naturally self-fertilizing, and when people breed them, I think that they don't really focus on the F1. They just self them down into recombinant embryolines.